our town hall for the fall of 2022. We are focusing on connecting science and stakeholders, looking at acidification in mangrove systems. And so we've touched a number of topics in the past, and we're super excited to look at the world of mangroves in this town hall. So I'm going to go ahead and start with some meeting logistics. So all attendees should be in listen only mode. If for some reason you find that you are unmuted, please go ahead and mute yourselves. But we, the moderators, will also keep an eye on that to keep you muted throughout the presentations. You can go ahead and enter questions in the chat for our Q&A session, and note which presenter you want the question directed towards. We will have 2 short presentations followed by a question and answer session. And so we will have time at the end. This is meant to be a discussion at the end, so we'll be able to all chat together. We are also recording this meeting. There are a number of people who have signed up, but were unable to actually attend live today. So I do know there's been some requests for the recording. We will post it to the Socorro YouTube channel, and we can guide you in that direction as needed at the end of this. And so I want to go ahead and start with just a brief introduction to who we are, SOCAN, and the virtual network. So SOCAN is the Southeast Ocean and Coastal Acidification Network, which is one of a number of CANs throughout the United States. We do represent the Southeast region, which is North Carolina, South Carolina, Georgia, and Florida, and it's the entire state of Florida. So we do overlap with one of the other CANs, GCAN, the Gulf Coast Ocean and Coastal Acidification Network. Our team is made up of Deborah Hernandez, who is the executive director of Socorro. So Socorro is SOCAN's fiscal home, so where we are based out of. We also have Laura Corman, who's a Socorro liaison, on the call with us and part of the executive team. Myself, I'm Dr. Emily Hall. I'm one of the co-coordinators of SOCAN, and I'm also a senior scientist and program manager at Mote Marine Laboratory in Sarasota, Florida. And our other co-coordinator is Dr. Janet Reithmaier. She is at the University of Delaware. Both of us work directly on acidification issues. We're both chemists in our field, but collaborate all throughout the Southeast and all throughout our region to try to understand what's going on in our region with acidification. Part of SOCAN includes 2 different working groups. We have a science working group and a stakeholder working group, which both meet every other month, alternating months from each other. And our stakeholder working group is made up of a number of stakeholders throughout the Southeast that have an interest or in either understanding or participating in understanding how acidification may affect them. And then our science working group helps us to write proposals, connect with the stakeholders, and get the message across. The history of SOCAN, we were formed in February of 2015, so we've been around for a little bit, but in the world of research and outreach, that's actually a really short time period. We do make up a network of an interdisciplinary group of scientists and stakeholders working together, which has been really productive for us so far. We do have a paper that was published in 2020, which was a collaboration of scientists and stakeholders. I don't have it listed on the PowerPoint here, but that is something that we can get to you. It is on our website as well. Our main goal is to gain a better understanding of the causes of acidification so we can work to reduce its impact in our region. There are many different influences of acidification in many different regions. And within the Southeast, even throughout the Southeast, we have different regions that have different impacts or different levels of acidification happening. And so I'm going to go ahead now and pass it over to Janet, who's going to introduce our 2 speakers who we are so grateful for having joined us today. So Janet.
Good morning, everyone. Thanks for joining SOCAN today. Emily, if you could go to the next slide for me, that'd be great. So I'm going to introduce our speakers. Our first speaker this morning is Dr. Gloria Reithmaier, and I hope I got that pronunciation right. Dr. Reithmaier received her PhD from Southern Cross University in Lismore, Australia. She is doing a postdoc right now at Gothenburg University in Göteborg, Sweden. Hope I got those right too. Her research focuses on carbon cycling of blue carbon ecosystems, including mangroves, salt marshes, and seagrasses. She investigates carbon storage, greenhouse gas exchange, as well as alkalinity and carbon outwelling. Although mangroves cover only a small area globally of less than 0.3%, they play an important role in the global carbon cycle. Large shares of terrestrially derived carbon are stored in mangroves or pumped through mangroves before entering the ocean. Mangroves are supposed to deliver alkalinity, primarily organic alkalinity, to the ocean buffering capacity against acidification. However, organic alkalinity production and export in mangroves is so far poorly understood, which is why it's so important that we start this conversation on mangroves. Our next speaker for today will be Dr. Cyronak. Dr. Cyronak is transitioning right now as an assistant professor from Nova Southeastern University to Georgia Southern University. So if you need to get in touch with him, you can email us through SOCAN and we'll pass it on to his temporary email address. Dr. Cyronak is a biogeochemist studying how material and chemical elements are cycled through marine and coastal ecosystems. His research focuses on the carbon cycle in systems such as coral reefs, mangroves, seagrass beds, and calcium carbonate sediments. He is interested in the role that these ecosystems play in the global carbon cycle and how they will be affected by climate change. Most of his work involves making seawater chemistry measurements in the field using state-of-the-art technology with a focus on carbon dioxide, methane, total alkalinity, oxygen, and submarine groundwater discharge. So we're going to let our speakers give their presentations, and then we will, as Emily said, open up a panel discussion at the end. So you can ask questions directed towards any of us. So Gloria, please go ahead.
Yeah, thanks for inviting me today. I will talk a little bit about the benefits of tidal wetlands that focus on mangroves, and especially how they mitigate climate change and how they impact coastal acidification. Yeah. So mangroves are big atmospheric carbon sinks. They store a lot of, so they capture the carbon from the atmosphere and store it in their biomass and in their sediments, and that's why they can help mitigate climate change. And if you look at the, as I already said, they have only a small area globally, but if you look at how much carbon they store per area, the carbon storage is actually much higher than compared to other forests, like for example, rainforests. And so far, the research has mainly focused on carbon burial in the sediments in mangroves, which is called blue carbon, and also includes the carbon stored by salt marshes and seagrasses. And when you look at how much carbon is captured and stored in mangroves, you also have to consider what happens to this carbon. And only a part of this gets actually stored in the soils. Some part of it is gets expired again as CO2. And also organic carbon can be exported to the ocean, which is called outwelling. But still accounting for all these parameters, half of the carbon budget was still unaccounted for. And that's what I and also my group are researching. So we don't only look at the carbon storage in the soil, but also the greenhouse gas exchange and also the lateral export to the ocean. So one of my studies was in the Everglades. Maybe some of you have been lucky enough to go there. It's a really amazing place. Probably know it's a national park, and it's protects the biggest continuous mangrove forest in North America. So it's a really amazing place. And on the right, you see how we went on a houseboat, which sounds very luxurious, but actually it only consists of measurement devices, like no bathroom, no toilet, no kitchen, no bed, just a tiny tent on the top, but still an amazing experience to go there. And we did a carbon budget for the Everglades mangroves. So in the northern parts of the Everglades, we have freshwater marshes that also input some carbon. And then we looked at how much is going out to the ocean, the greenhouse gas exchange, and the carbon barrier. And when you look at the pie chart, which is basically the same we already had a look at, but specifically for the Everglades, we found that, yeah, carbon barrier accounts for less than 1/4 of the carbon produced by mangroves. And actually the carbon budget is dominated by inorganic carbon export. And inorganic carbon, you have to differentiate when you look at how it actually impacts climate change mitigation potential as well as the pH. Because inorganic carbon, so DIC is dissolved inorganic carbon, consists of CO2, which is basically the bad guy. So it forms carbonic acid, which then acidifies, but also it can outgas back to the atmosphere, which we don't want because we want the carbon stored permanently in the ocean. But also DIC consists of alkalinity, so carbonate alkalinity. And this is the good guy, like it buffers acidification, and also it stays dissolved in the ocean for millennia. So it's basically a long-term carbon sink. So during my PhD, I focused on alkalinity production export in mangroves. And alkalinity, but also DIC are both produced in the sediments in mangroves. And it's all linked to carbon decomposition or respiration. So like sulfide reduction associated with pyrite burial, for example, is the main permanent alkalinity production process. But also we have organic alkalinity, but I have to make a correction there from Janet. It's not the main alkalinity. The main alkalinity is carbonate alkalinity, but organic alkalinity can have also some, can contribute in mangroves because they're so carbon rich, and organic alkalinity is associated with organic acids. And then, for example, in the Everglades, we have a carbonate basin, and carbonate dissolution that is facilitated in the pore water mangroves can also produce alkalinity. And for, so we have this alkalinity enriched pore water in the sediments in DIC, and this is then exported to the surface water by tidal pumping. So during the flood tide, the water comes in, infiltrates the sediment, basically is pushing out the old pore water. And this is facilitated by crab burrows, for example, or old roots. And then it's finally exported during low tide or ebb tide to the ocean. And as I said, yeah, it's very important to say, is this DIC, you know, is it like CO2 or is it alkalinity? Because yeah, it definitely makes a big difference. So what this is my latest project, which I just submitted yesterday. So you're the first to actually see this data. This is like a global review for mangroves and salt marshes. So we got a lot of data sets for alkalinity and DIC production, as well as outwelling globally. And there we look at the ratio. So you see the mangrove pore water, and then also surface water, and salt marsh as well. And for in pore water, we have a ratio of around 0.8, and it's the same for the outwelling. So basically, we have more DIC production and export than alkalinity. And this means that, yeah, it acidifies. Yeah, next. As I already told you, because we have the CO2, which can form carbonic acid, but still it's how it actually affects the pH. It's not that trivial to say from this ratio. It's just that when it's below 1, it acidifies, and if it's above 1, it would buffer coastal acidification. Yeah, but looking at the pH, it's also not that surprising, because like most terrestrial ecosystem actually acidify the ocean, and that's also because the ocean has really A lot of alkalinity, especially much more high alkalinity to DSC ratio. So it's kind of hard to top that and buffer the ocean, basically. So it's not as bad as it sounds. And also here you see the alkalinity to DSC ratio is increasing with pH, which is expected, but it's actually very scattered, especially around a ratio of 1, which is due to the minimum buffer capacity at this point. That's very theoretical, and I tried to break it down, but another way of looking at this is using the buffer factor beta H. And you can see that the pore waters in mangroves and salt waters have a much higher buffer capacity or buffer factor compared to surface water and as the ocean. So that means we can have a lot of DSC input, but the pH change is relatively small compared to this high DSC input. And another interesting thing we found is that we compared like pristine mangroves and salt marshes with anthropogenic impacted systems by urban areas or also farming activities. And we found that the pristine mangroves and salt marshes have much higher TA to DSC ratios. So basically they acidify less than the impacted sites. And also, as I told you at the beginning, is that it's not only the pH that is affected by this, but also the alkalinity outwelling. This is a global map I made with several different studies. Like this alkalinity is a permanent carbon sink from the atmosphere. So to sum up, yeah, alkalinity export from tidal wetlands is made like a permanent carbon sink that can help mitigate climate change, and also pristine wetlands like acidify less compared to impacted wetlands. So basically it's important to protect and restore mangroves and salt marshes in order to reduce coastal acidification and to maximize carbon sequestration. Yeah, and I want to acknowledge especially Damien Maha, who was my PhD supervisor, and Isaac Santos, who I'm working with right now, and then also all my co-authors who contributed to the data I just showed you. I think I saw at least one of them in the audience. So thanks for showing up, and yeah, thanks for your attention. And if you have any questions, I don't know if it's now or later, probably the time.
Nope, thank you so much. That was great. We're going to do questions at the end. And so I'm going to go ahead and pass it over to Tyler. So since Janet already gave a nice introduction of who you are, we'll go ahead and move right into your slides.
All right. Thanks, Gloria. That was really interesting. So today I'm going to sort of move on from what Gloria was talking, expand on a little bit, and think about how we can use some of this variability that occurs in coastal ecosystems and carbonate chemistry, not just mangroves. So here I've got seagrass beds, marshes, coral reefs, and how we can maybe tap into this natural variability when we think about adaptation, mitigation, and management of ocean acidification on a global scale. So next slide. So the first thing to think about when we think about changing global conditions is the difference between weather and climate. I don't think I need to harp on this in this crowd, but you know, maybe if I was giving a speech to the United States Senate, there'd be some people who need to learn this difference, right? So weather is really kind of the conditions at any given time. You look out your window and you see it's raining or sunny, that's weather. Climate is a statistical representation of weather, usually over 30 years. We look at the averages and we see how those change. Next slide. A really great quote that sums this up is by Robert Heinlein, who's a science fiction writer, is climate is what you expect and weather is what you get, right? Climate is this expectation over longer periods of time. And just like in the atmosphere, we have these sort of ideas of what we're experiencing with the state of the atmosphere, right? The same thing happens underwater. Next slide. And we can think of these same things occurring in seawater or the ocean, right? We have ocean weather, which is maybe the state of the seawater at any given time. And on this slide on the left in the white, you can see temperature variability on a coral reef at 3 different locations. And you can see temperature over the course of a day, these are daily cycles that you're seeing there, is swinging anywhere from 20 degrees to 28 degrees Celsius. So an 8-degree swing almost on a daily basis. So these organisms are experiencing sort of similar weather that we experience in the atmosphere, except with the ocean. And there's also ocean climate, right? And these are the averages over extended periods of time over different regions of the ocean or over the whole global ocean itself. Next slide. So one way to think about this is we don't really appreciate what the organisms in the ocean are experiencing because we obviously evolved on land, right? So we're used to atmospheric weather and climate and experiencing that. But these organisms actually experience very similar things. And if you've ever seen this movie, it's a great movie about a guy who goes snorkeling with this octopus every day for an entire year and really gets to sort of interact with this really highly intelligent creature. If you haven't seen it, I highly recommend it. But there was one quote in there that he says that really made me think about this concept of atmospheric and ocean weather and climate. He says, it took going in every day to really get to know her environment better. And what he was talking about was the changes in the state of the seawater there. So some days there were big currents, some days it was really turbid and he couldn't see very far. Some days it was cold, warmer, things like that. And it was really these very dynamic changes in the seawater itself that he was referring to with this quote. And this has sort of made it into the scientific literature. Next slide. There was this opinion piece in Nature talking about how we need to think more generally about ocean weather, these changes that these organisms are experiencing, and the conditions that they're seeing over various time and spatial scales to think about how global climate change is going to impact not only their habitats, but their environments and maybe their ability to adapt and to withstand climate change as a general sort of global change. Next slide. So I want to sort of talk about this paper that Lydia Katzenberg and I wrote in 2019 in Global Change Biology. And really what we were trying to think about is how can you use ocean weather to potentially find ocean acidification refugia, right? These places where ocean acidification might not be so bad. You can also flip this on its head and think about how we can find places where ocean acidification might be made worse by some of these dynamic natural changes that we, can generally call ocean weather. And on the graph here, you can see different spatial and time scales of changes in pH across different ecosystems. So we can have seasonal scales due to just changes in solar irradiation over seasons. We can have bigger sorts of changes on upwelling and phytoplankton blooms, which we can maybe call regional processes. And as we get closer and closer into the organisms themselves, you get down to the point where really the boundary layer around the organism is actually having quite a bit of variability in pH, where depending if that organism's calcifying or photosynthetic, you might have really low pH close to that organism compared to a few millimeters or centimeters away. And then on the bottom there, you can see different examples of how pH might vary over these different time scales from seasonal to daily changes in pH. And one thing to bring your attention is that some of these changes, especially happening on daily time scales in places like coral reefs or weekly regional process where there's upwelling, are sort of dwarfing the magnitude of global ocean acidification. Next slide real quick, which we can sort of see here. So these changes in the global open ocean pH, we can see in that blue there, it's going from about 8.13. This is pretty old data here, so not all the way up to 2022, but you have about a 0.01-ish decrease in pH, 1 pH unit in some of these ecosystems. And so how do these global changes like ocean acidification, right, which is due to the atmospheric increase in CO2, interact with these local, regional, and smaller scale changes? That's kind of the question we were after. And what we did was we dug through the literature and looked at places where scientists had done experiments to sort of test how the variability will impact this change in average conditions. Next slide. But before we did that, we had to sort of think about how to frame this. And really, this is a paper from 2011 thinking about how organisms and biodiversity are going to change with changing climate and how conservation should really handle this, conservation initiatives. And really, they framed it in this way, Dawson, as vulnerability. And vulnerability is the extent to which a species or population is threatened by climate change. And there's three main components. The first component, next slide, is sensitivity. And this is really the degree to which a species or population is dependent on the prevailing climate. So if it goes outside of the natural averages of that area, how does it affect them? So next slide. Adaptive capacity, and this is the ability of a species or population to cope with climate changes. So this can be phenotypic genetic capacity to adapt to these changes, or maybe they're shifting ranges, migrating, things like that. Next slide. And exposure, and this is really what I focus on in my research, which I'll show some examples about in a second. But really, this is the extent of climate change likely to be experienced, and this really depends on that natural state of the ecosystem and how the natural processes are changing pH on different time scales. And that's going to influence ocean acidification trajectories in those individual ecosystems and locations. So this concept of vulnerability is sort of three-pronged, and two of them are really based on the organism itself, right, or the population. And then this other one, exposure, is really based on sort of the weather, right, the environment they're experiencing. Next slide. And so what we did was we divided up the experiments in the literature based on ocean acidification. So we were interested in ability in pH and natural ocean, and we divided up into two sort of categories. Environmental history, which was where scientists took organisms from an environment with variable pH and an environment with more stable pH, maybe upwelling versus upwelling shielded area. And then they did experiments to see how ocean acidification affected various physiological functions. And then there was direct exposure, and this was laboratory-based experiments where scientists would take an organism and put it in a very stable condition, like constant seawater pH, and then maybe put it in a lower pH that's stable, but also have treatments where they've got variability over different time scales within those experimental tanks as well. And so this was testing, does variability itself impact their response to a similar mean reduction in pH? The overall things we found, so here in the bar graphs, you can see positive effects. So these were statistically significant positive effects on organism physiology. Negative effects were bad things that would happen to the organism. So maybe reduced growth, reduced reproduction, things like that. And zero was when there was no significant effect of any of these treatments. And overall, variability really often doesn't matter so much. Most of the time, it pretty much, especially with direct exposures, had minimal impact on the physiology of the organism. So what we really took away from this was that reducing CO2 emissions remains the primary management goal. And that's something that you hear a lot, especially with carbon dioxide removal now, right? We still have to reduce CO2 emissions. That's the most important thing. When variability does matter, things are a bit inconsistent. So it's not necessarily a completely reliable solution, but there was this sort of green light in the left-hand side there. You can see previous exposure to low pH and variability might be able to enhance OA tolerance. And this is maybe organisms living in upwelling zones that already experience high levels of pH variability. Maybe they're a little more adaptable to ocean acidification, and maybe they can handle the changes those populations. So we thought about this in the idea of refugia. So next slide. And refugia are sort of these places that might be able to mitigate ocean acidification. And like I said earlier, you can sort of flip this and think about as hot spots as well, places where ocean acidification could be made worse. But really, this is showing you in graph form the idea of these two different types of experiments. We looked at these changes in the pH from a constant mean to high variability, to this environmental history kind of experiment where organisms were taken from different sites and sort of transplanted across them. And we came up with a few types of refugia that managers and people might want to think about when it comes to trying to mitigate ocean acidification. Next slide. And really, there were sort of three that we came up with. Spatial refugia, and these are areas with less intense OA exposure relative to sister populations. So maybe upwelling shielded areas where there's the Channel Islands, or in Southern California are a good example, where some of the islands are exposed to a lot of upwelling, some are not. Small scale operative refugia, and this is really what got us started thinking about this whole process, was this idea of phytoremediation, or using plants, seaweeds, and seagrass to try and reduce the impacts of ocean acidification by drawing out CO2. And these might work in some cases, but you have to really, really be targeting a specific organism and know that organism is going to respond beneficially to this change in mean pH. And it also requires significant investment, right? Like you have to really be putting a lot of effort into that compared to the spatial refugium, which might just exist naturally. And then the sort of refugia based on adaptive capacity, where the exposure isn't necessarily the thing that's important, but changing that organism's adaptive capacity through different exposures might be the thing that is driving that. So these were all sorts of ideas that we came up with as far as the types of refugia. And it all comes back to the organisms, what the organism's experiencing, right? Next slide. So we can go back to this and think about the organism, this octopus, and what it's experiencing in the movie, and how its environment is changing. But the real question is, how do we go in every day to measure these changes? So how can we go and find these places and understand what's happening naturally to take advantage of this variability that might be leading to enhanced adaptive capacity or maybe reduced exposure to ocean acidification? And this is where I would normally talk to you guys about some citizen science projects I'm working on, but today I don't really have the time. So what I'm going to do is highlight two of my graduate students from Nova Southeastern, their work. And I guess how you go in every day, sometimes you have to send a graduate student out, right? And so that's what we'll talk about next. Next slide. I just want to highlight specifically a couple that we did in South Florida, a couple of experiments. So this is Alexandrina Rangel. She was working actually through the pandemic. The pandemic hit in the middle of her trying to get out and do some field work in these little mangrove estuaries in South Florida. So Port Everglades, which you can see sort of highlighted on the top of the left map on your screen, is one of the biggest ports in the United States, big container ships going in and out. But just south of there, there's actually some mangrove forest that is still kind of healthy and maintained in these little state parks there. Otherwise, along the South Florida coast, there isn't much mangrove. The other side, there's lots of mangrove forest that's still there. But here it's kind of spotty and hard to find from all the development. And so what Alex did was she went out into these two mangrove environments, West Lake and Whiskey Creek. One was sort of on the ocean side of the intracoastal waterway. The other one was on the landslide. And she took measurements of carbonate chemistry and some other parameters, data with that. Next slide. And the main takeaway was that alkalinity and DIC, so this sort of buffering capacity of alkalinity, right, and the carbon, was really related to the salinity. So it showed trends with salinity in that alkalinity was increasing in Whiskey Creek, and so was dissolved in organic carbon compared to the higher salinity. So as you went down in salinity, you had higher alkalinity and DIC concentrations. And actually both of them, if you extrapolated these lines out to the zero salinity line, the fresh water actually had higher alkalinity than the ocean water off of the coast here. And one of the main takeaways from this, and if we think back to some of the things Gloria was saying about TA to DIC ratio and how that really is what controls the pH of these ecosystems. So a higher ratio means more buffering capacity. Seawater is about 1.15, 1.2. Anything below that is going to sort of reduce the buffering capacity as it mixes with that seawater, right? And what we can see is that salinity is really driving this buffering capacity. So as far as sort of tying this into refugia and hotspots and thinking about how we might be able to manage ocean acidification more regionally, this was really just seasonally driven. Florida, you might not think of having seasons. It really only has two, a wet and a dry season. And so maybe there's things where the wet season is going to lead to higher exposures, right? Reduced buffering capacity, more acidification. And maybe there's something we can do there with managing water discharge into these estuaries to try and reduce that acidification. So that's sort of tying in this temporal change in chemistry. And then next slide. I had another student named Harrison Davis, and he took advantage of a Florida Department of Environmental Protection project, which was measuring nutrients in all these inlets. So Port Everglades is one of these inlets that goes from the ocean side into the intracoastal. And up and down the coast of Florida, he monitored 9 inlets, all the way up to Port St. Lucie and down to the government cut, which is down in Miami. And basically every month he would go out and take carbon chemistry measurements within the inlet in this sort of gridded pattern. So a little bit north, a little bit south, east, and then sort of in the middle of the inlet. And so we use this chemistry to sort of get an idea of, is there any variability in the buffering capacity of these inlets across this north-south gradient. So next slide. So we used the same sort of concept where we plotted salinity versus DIC and alkalinity. And because we didn't really have the capacity to go out and measure the fresh water that was being discharged, which is most likely a combination of both surface water from Lake Okeechobee that's moved through the Everglades and is moving from Lake Okeechobee, which is up by the northernmost inlet down south, and groundwater discharge as well that's been moving through the sort of calcium carbonates, karstic bedrock there. And because we didn't actually have measurements of that fresh water, it wasn't really in the scope of the project and funding, we use these relationships in each estuary or each inlet to back calculate what the zero salinity end member might be for both alkalinity and dissolved inorganic carbon. Next slide. And we found this neat latitudinal trend, which we really need to investigate a little bit more. We really need to go out and measure the groundwater and the surface water itself as it's moving down the coast here. And what we found was that the alkalinity and both DIC end member increased as we went further south. And my sort of guess as to why this occurred is that as the water is moving from Lake Okeechobee, so really Lake Okeechobee is the headwaters of the Everglades, it spills over and it just slowly drains down south through South Florida. But this is heavily regulated as far as water movement goes by the government, because they're, actively moving water in different directions to avoid flooding and to irrigate crops and things like that. But in any case, the residence time increases of that water within the terrestrial side increases as we go down south. And so I think that's somewhat related to why we're seeing this increase in alkalinity and DIC as we move further south. And really, this is another sort of, to tie this back into refugia and hotspots of ocean acidification, we can think that the southern north to south gradient is actually going to have different ocean acidification trajectories, potentially because of the interaction between the terrestrial derived freshwater and the coastal seawater. And in these ecosystems like mangroves, like coral reefs and seagrass that are actually inhabiting this sort of area between ocean and land, this is going to be places where maybe we can target different strategies to think about, if we modify flow, do things like that, or we know areas are going to be hit harder by ocean acidification, we can think about how to handle that a little bit differently. Next slide. So really, I just wanted to highlight this framework for thinking about variability when we think about ocean acidification, especially along the coast. The open ocean is way more homogeneous. The coast is very dynamic. And this variability could create ocean acidification refugia and hotspots, and maybe we can use this to our advantage. So we need ways to measure carbonate chemistry at higher spatial and temporal resolution. So those projects were, 2 master's projects, and the students did a lot of work. They were out there a lot taking these samples. So it's really hard to find ways to measure things both across space and time when you're looking at these big areas. And so we need to think about new ways to do that. Maybe citizen science is a way. Sensors are obviously a nice thing, but sensors tend to be expensive. But different ways to monitor the spatial and temporal variability. And when you're doing experiments or when you're trying to monitor things, one thing to think about is what is the important spatial and temporal variability I need to think about in my specific experiment. And then thinking about the ocean weather or thinking about ocean weather that organisms experience can help us sort of harness this variability and combat global change. So thinking about what the organisms, like what that octopus is experiencing, is in the same sort of framework that we think about our own weather, right, might help us to understand what these organisms are experiencing and how it will impact undersea organisms, just the same way weather and climate impact above sea organisms. So that's all I have. I just want to thank all the collaborators, especially Lydia Kapsenberg, Alex Rangel, and Harrison Davis for their help with all of these experiments and sort of meta-analyses, and I'm happy to answer any questions.
First, we wanted to say thank you to both of our speakers for joining us today. I know I learned a lot about mangroves, especially what I don't know. So thanks again. If anybody has any questions, please go ahead and write them in the chat. There are a couple that are popping up already. So our first question is, and this question came up during Gloria's presentation, so I think it's probably directed towards her, but this is a panel discussion, so we invite all of our speakers to weigh in. Do you know what it is about mangroves that allows them to store much more carbon than terrestrial forest or trees?
It's basically like the anoxic environment of the sediments. So when you have oxygen, the bacteria, like the dominant bacteria, just respire all this carbon back to the atmosphere. But we also have carbon respiration, but the anoxic one is much slower. And it's kind of like with food, you know, you can eat chocolate, you eat it quick. And that's basically the oxic or the aerobic respiration. Or you can eat, I don't know, your least favorite vegetable, and maybe you eat it a little bit slower, and it's kind of stays in the fridge for longer. And that's with the anaerobic respiration. It's just that the water is most of the time over the sediments and it's waterlogged, and that's why it just stays there. And then also we have this additional carbon that goes to the ocean. With rainforests, they're not connected to the ocean, it's just atmosphere or soil. So it's basically a different hydrology.
Awesome, thank you. Tyler, do you have anything to add to that?
No, I think that's, exactly right. The lack of oxygen is just allowing that carbon to be stored the same way, our oil reserves were stored in the deep ocean, and we're pulling it back out and dumping it in the atmosphere now. But also, I think mangroves are very efficient at trapping organic carbon too. So it's not all necessarily produced by the mangroves themselves, but the coastal flow of carbon through rivers and streams eventually makes it down to them, and their properties just allow it to be trapped, sort of settled out.
Interesting. Another question that we have is, in situ observations are limited in locations and time. Can coastal ocean numerical models be used to fill in spatial and temporal variations of salinity, TA, DIC, pH, etc.?
I think they can. For example, I tried to model like alkalinity and DSC export from mangroves globally, but it did not work with the given data we have. Like there's just not enough clear global trends. And as Tyler said, it's like the seasonal changes and stuff, and all this stuff is very hard to measure. So usually like this outwelling rates are based on only 2 tidal cycles, so it's like a snapshot in time. So it would be much better to have like this long-term measurements where sensors can be used. Like alkalinity, it's just a problem with the sensor. If you, for example, there's no alkalinity sensor, so you would have to measure like CO2 and then calculate alkalinity, but alkalinity can also be part that can be organic, and this is then not part of this sensor measurement. So it's a little bit more complicated, but it can be a proxy, I think, if you use that.
Yeah, I agree. I think, the models are really great, but they need to be verified, and, they work well in the open ocean. The closer you get to the coast, you have such dynamic things happening, and especially when you start to consider the input of things from land. You could go from one estuary just like a degree south to another estuary, and things are totally different because of the watershed. And yeah, so it's just a really tough thing, but I think they definitely play a role for sure.
I have a question that I think might tack on to that. Given that climate models have predicted that the Southeast is going to get wetter, how do you think that that might affect the overall buffering capacity within mangroves, but not just within the mangrove itself, but maybe within the sediments as well, as far as increased groundwater, or just maybe like an increase in oxygenation of the sediment? Like what kind of, Tyler was starting to talk about like the weather versus climate. What kind of climate predictions do you think you would see as far as acidification goes in mangroves?
I mean, I think that's going to be such a complicated question, because once you start to change the climate on land of these places, right, and you're changing precipitation patterns, you're going to change groundwater inputs, you're going to change surface water inputs, you're going to change the amount of weathering that's happening, the amount of organic growth on top of the soils where this water's coming from, and then eventually making its way back to the ocean. So I don't know that. I mean, I think the answer is definitely there is going to be an impact. I just don't know what it's going to be, and whether there is going to be one sort of global answer, right? Like, I think it's just going to be so different from every different watershed and different ecosystem that it's going to be really tough to generalize.
Yeah, I think so too. That's not, sorry.
Oh, go ahead, Gloria.
So it's yeah, the easy way to say, yeah, just the temperature rises, and that's climate change, but that's not how it works. You have like sea level rise that impacts, and this extreme weather events. So you can have a cyclone which can have a big impact, and then just a few kilometers away it's like a rainfall or drought. Or so I think there's not one answer to this, except for it's going to change.
So then, would you guys agree that managers such as the managers from National Estuary Programs, NERS, and other government bodies, would those managers need to take into account acidification when thinking about mangrove and seagrass restoration and recovery?
Yeah, I think they can take it into account. I think at this stage, it's a very theoretical still, because there's not too much known about it. But what is for sure is that like mangroves provide so many ecosystem services, like the carbon, climate change mitigation, their nurseries and so on. So like there's so many benefits that you don't have to focus on this one thing, like an acidification, but it's just in general safe to say that they should be preserved and restored and protected.
And I think mangroves might play a big role in helping to buffer acidification in certain areas if the alkalinity being released is high enough that the alkalinity to DIC ratio is actually helping to buffer that coastal system. And mangroves are in close proximity to other ecosystems that are really highly susceptible to ocean acidification, like coral reefs, for instance. Seagrass might actually benefit from a little bit of acidification because it might enhance their primary production a bit, but it's really complicated at the ecological level, because then there's all these other feedbacks and organisms that live in these ecosystems that might not benefit. But yes, I think it's definitely something that needs to be taken into consideration, and especially thinking about this renewed interest in ocean alkalinity enhancement as a way to combat global climate change and carbon dioxide, to draw down carbon dioxide from the atmosphere. I think thinking about how these systems play a role in that is really critical. I think we need to, the global alkalinity budget has a pretty big hole in it, and we kind of need to close that and figure it out. And it's just a really complicated thing to think about.
So you're starting to talk about refugia and enhancing the alkalinity of coastal waters. Have you seen, or have there been any studies yet that have identified species that have maybe started to migrate into mangroves that weren't historically found there?
I don't know of a study, but I know from sort of looking online and some news articles that mangroves have almost made it out of Florida across the Georgia-Florida line. So mangroves themselves are migrating, and they're going to start, you know, as the temperatures allow, might be taking over some of this marshland that was there. And so now the question becomes, is that ecologically beneficial? You know, how does that change the carbon carrying capacity of these coastal systems? So I know that's happening. And then for sure, coral reefs, there's been evidence that coral reefs in Japan are migrating north.
Wow.
At least coral species, maybe not the reefs themselves yet.
Well, it does take a while. Another question we have from the audience is, how do you measure the parameters of the carbonate system when the water is full of sediment or organic matter? Sounds like a technical question for field.
So yeah, there are different methods and they're all complicated. So the easiest one is obviously to take to measure these parameters in surface water, because you can just get a vial full of water. In pore water, it's really quite hard to get. Like, one thing is digging, just dig a hole, and then, you know, you need to filter and so on. But we also try to get like big sediment cores, and then you have like, it's called like a rhizome. So you stick in little filter thingies, and then you can filter out, but it takes a long time and you just get very little water. But because you have so high, like so much alkalinity in there, it's still enough to measure. But it's yeah, very labor intensive work, I would say, in general in mangroves, because, you know, like the mud, and you just sink in until your hip, and it's very hard to walk. And I think many people have this very romantic idea of marine researchers, I don't know, snorkeling in pretty water, but actually most of the time you smell and you're dirty and it's really hot, and the mosquitoes, and you wear full body protection. So it's, yeah, it's quite challenging, I would say.
Crocodiles sometimes.
Exactly. Yeah, after 3 years in Australia, I'm still a bit scared to go in the water if there are mangroves around. So in the Everglades, I was very hesitant to actually jump in the water.
So we have a comment from one of the SOCAN members that's from Michael Leah, and he says that, and I would love to draw everybody's attention actually to the chat, because there are probably several folks on this webinar today that also study mangroves, so not just our speakers. But Blue Ocean Sails would welcome the opportunity to collaborate on development of its citizen science carbonate chemistry measurement study. And you can take down Mike's email there. It's michaelia@hotmail.com, which, if we don't have any more questions coming in from the audience, then I will kind of start to transition to our wrap up by saying SOCAN is here to help with connecting members and researchers across the Southeast. We also do a lot of work with GCAN over in the Gulf of Mexico, and we're hoping to expand and do a little bit more work with MACAN, which is the Mid-Atlantic Coastal Acidification Network that's just to our north. So we're here to help facilitate research and proposal writing and connecting members and folks across the region and to other CANs as well. So if you have projects that you'd like to collaborate on, SOCAN is a great way to do that. Emily mentioned at the beginning that we have 2 working groups. If you are interested in participating on one of our working groups. I think this month we meet the working groups meet every other month on the third Monday at 2 p.m. And it's pretty much open to anybody that is willing to participate.
Oh my God, you can't print it. Well,
So please go ahead and contact us at SOCAN@socorro.org. Or if you're interested in collaborating with any of our speakers today, I would hope that, or have questions for them, you can also email us and we'll be happy to put you in touch with them. Or I think they may have shared their email addresses on their presentations. I don't remember. So we thank everyone for joining us today. We're going to send out a link to this, to the recording of this presentation that'll go out in our next member email. And again, it'll be on our website. So please check that out at socan.socorro.org. And if there's nothing else from the speakers that they might want to add, then we will wrap it up for today.
Okay, thanks for inviting us.
Yeah, thanks for having me.
Awesome. It's been great, guys. Thank you so much.